Okay, thank you so much for this opportunity. I'm Kary Ocaña. I work in the National Laboratory of Scientific Computing in Rio de Janeiro, and this is an interdisciplinary work because involved informatics and also HPC. And then I'm going to introduce some concept of both of them, in general the transcriptomic, and then this is my team and collaborator and student that involves national and international collaborators involving RIA, Gottingen, and Universidade de Tomar in Portugal. And I am the only bioinformatics here. But okay, the thing is that in each experiment of bioinformatics, we need to model the experiments, and we need to thinking about the probably bottlenecks that we need to better study. And in this case, for example, we have a lot of data and we have the software also, and we have the different type of environmental computer system that we can use. But why we study transcriptomics? Because transcriptomics in informatics is one of maybe the only area that is considered as biological big data, because it generates and consumes a lot of huge quantity of data. And we process all the executions in this study was processed in the supercomputer Santos Dumont that also is hosted in our institute. This is a little introduction of the RNAseq, the transcriptomics workflow, but the more important thing is that in transcriptomics, the difference between the genomics and transcriptomics is that in genomics we are going to identify some genes and the function of that genes, but in transcriptomics also we are going to relate these genes with the quantity of the genes, and we are going to study about which one of the genes are expressed, overexpressed, or downregulated. That is very, very important for constructing new machines, for example, or for study for discover new drug target. The thing here is that we are going to do some comparison between the quantity of the genes that are expressed in controls and the quantity of genes that are present in some pathological or some physiological conditions. In this opportunity, the data was generated by the unit of the toxicology of the University of Gottingen, and they use little mouse, and the focus of this study was what is the difference between the expressed genes between the adult mouse, cardiomyocytic mouse, and the baby's mouse, and then if there is a difference, because there is an hypothesis that in baby's mouse there is a gene that maybe protects for any problem of any cardiopathology, for example. And we know the laboratory extract this data and compare. And my work here was in the third level, that okay, we needed to model the experiment, we needed maybe process using HPC, and also when we obtained the results, we sent for the specialist that is going to the group is composed by biologists or bioinformaticians, but our work here is only the executions and the modeling of the workflow. This is the initial modeling. I remember that the execution that the colleagues, the workflow system that they use in this Galaxy, and then only they execute them by website, and then there are a lot of problems, and then we propose that we can install and deploy all the bioinformatics software and all the bioinformatics libraries in the Santos Dumont, and also we model the workflow, and we study which one is the bottlenecks related to the software that we can optimize to model this workflow. This is for one patient, for example, but the idea and with the optimizing workflow is that we can explore in a lot of patients, and then we explore and we obtain the data, improving the performance in the executions. Okay, we starting with, okay, if we have different type of softwares and then we know the code of the software, then we can obtain the information of which one are the softwares, for example, the Boito, the first one, or the Santur, the second one, that we can use multi-threads for the execution, and then, okay, this is a point that we can optimize. And the second one, for the RNAseq, for example, here, is only not present threads, but present different process, and then this indicates that we need to split the data to introduce this data to these programs, and then after that, with that information, we modeling the workflow, and we started with the executions. The first version of this workflow only present 3 activities, but we needed to insert, or we needed to modify the workflows, and in this case here, we started with some initial execution to identify, okay, this point, this is a bottleneck, okay, we annotate all this information to explore more about this software. In here, for example, this is the first version and this is the second version, and the idea is that the second version also, the results of the performance are improved in comparison with these previous versions, but the difference is that the version or the program use different type of version of programs. The biological data need to be the same, because we are working with real data, and the variability and the reproducibility of these workflows must not vary the biological data. And okay, for example, for Boito, it was very nice, because we know that this software, this activity, use threads. But as we model it with the Parson, that is a language that we use to model this workflow and to manage the workflow and to execute to Santos Dumont, also have the threads. And then we also, it's obvious that when we combine the threads that are called from the software and the threads that are called from the workflow systems, we obtain the better performance here. And with the other information here is that not is necessary to use the 24 cores, because the best performance is here in threads, using these threads. And then this optimization was included step by step in the workflow. The second improvement was like I said, we needed to split the data here. When we include two activities, but the thing is here is that with that we obtain 90% of improvement only in here, and in the total execution time we obtain at the total 70% of the improvement in this performance. We also, when we explore the CPU, we thinking about, okay, but what if it's a biological data? I know that it's probably the consume of the read and write also is significant, and we obtain that information using Darsham that collect this information from the Santos Dumont, and also for understand about that the softwares in bioinformatics has different profiles. For example, in this case, these are the most representative activities, and this is the largest file, and this is the smallest files. In Boito, there's no difference, because but the 90% was related to grad operation. But in Solve, for example, there is, if we include the size of the data, we're going to the difference here from example with the smaller data, the other operations are more significant than the write and read. But when the size of the data is increasing, the read operation also are increasing, and this can be a problem if we execute a large scale this type of workflows producing a huge volume of data. And when was with that, all this information, that this is all our last computational performing here is okay, but we don't need to move the data from the ECCD and Lustre, and then we have the input in the Lustre, we are only moved once in the ECCD, and we are going to execute all the activities, and only the final execution, the final data is going to be moved in the Lustre to execute the last activities, and it's working well, because with all of the massing workflow we obtain here with this modeling, an improvement of 10%. Here is for our group in bioinformatics in my laboratory. This is a very important thing, because we can. I want that you observe that the biological data is not different between the previous and the current ParslRNA-Seq execution. It's the same, and then the reproducibility is nice, and the same final data was obtained, and then it's nice because it's real data. And from that, for example, the important thing is in the point of view of the bioinformatics is that yes, we obtain genes that are differentially expressed here in red, okay, and then it's probably that if we compare the control and if we compare the pathological condition, it's a difference, and then we needed to see which one are the genes that are non-regulated or are overexpressed, and it's probably that this difference maybe needed to go to the laboratory and to do the RNA sequencing, etc. But this is with the in wet laboratory. And the second one is my final result here is that in here we have the thousands genes that are more variables, and we can observe that here in red are the overexpressed, and in blue are not regulated, and we can see that are difference between them, but we are working only with cardiomyocytes, and the cardiomyocytes are in specific tissues and in the heart, and from these 100 genes is only 10 genes that we are going to work with them. And then this is other work, and it's probably that these genes need to be returning to the wet laboratory, and maybe the workflow need to be re-executed, and etc. Okay. Well, my conclusion is that we obtained we call it an optimizing workflow, and our focus was the optimization of the workflows. Okay, the most important thing here is our biological data is the same, and then we explored the most metrics, computational metrics variables that in CPU or in the executed in different architectures or in I/O, and now is executing in large scale for that. Okay, thank you so much for this opportunity.